Next one is nomenclature of the enzymes. Okay, after the discovery related to the various types of enzymes, now the naming of the enzymes is also very much important and vital to identify the enzymes in which they are participating or in which type of reaction they are participating. So, in that one there are many criteria are there for naming the enzymes. So, let us take the first criteria related to the suffix ase is given to the name of the enzyme. For example, if any enzyme is there or any enzyme is there, that enzyme have to be ended with a ase, like depending upon the type of the substrate on which it is acting, like if it is acting on maltose compound, so here the name of that enzyme is a maltase, that is it is ending with a ase. If it is acting on sucrose molecule, so the enzyme is called as sucrase enzyme, like it is ending with ase. Likewise, every enzyme is named by adding ase to the ase as an suffix to the name of the compound or the substrate on which the enzyme is acting. And next one is based on the name of the type of reaction. The second criteria for nomenclature of enzymes is a name of the type of reaction in which the enzyme is participating. For example, if the enzyme is participating in the dehydrogenation reaction, so we call this as a dehydrogenase enzyme. So, here also at the end of the type of reaction it is ending with ase or suffix ase is used. And if it is participating in the isomerization reaction, then the enzyme is called as isomerase enzyme. Here also it is ending with ase. In the same way, it is if the reaction if the enzyme is participating in a carboxylation reaction, so here the enzyme is carboxylase. Here also it is ending with the ase. So, likewise in the first and second criteria, the ase is the suffix which is used here, but in the first criteria at the end of the name of the substrate molecule, but in the second criteria it is at the end of the name of the type of reaction. And the third criteria is the here substrate, the type of reaction and ending with ase. All the three are considered here. So, that is, for example, if any compound, any substrate is participating in some type of reaction, like malate is a substrate molecule which is participating in the dehydrogenation reaction. So, we call it as malate dehydrogenase. So, here malate is the name of the substrate and this dehydrogenation is the type of reaction and that is ending with the ase. So, likewise glutamate synthesis, or so we call it as glutamate synthetase enzyme. So, likewise here the type of the compound, the type of substrate, the reaction and the ase. So, likewise the third criteria is followed, and the fourth criteria is arbitrary, that is there is no specific name or rule that have to be followed. Like let us take some of the examples like trypsin, pepsin. So, this type of enzymes neither they are having a name of the substrate nor the type of reaction nor it is ending with the ase. So, likewise different criteria have been followed. So, here different criteria have been given by the organization called as IUBMB, that is International Union of Biochemistry and Molecular Biology. So, likewise the criteria have been given for the nomenclature of the enzymes. Here next one is chemical nature of the enzymes. So here as we said that these enzymes are proteinaceous in their nature, but exceptionally as we said that ribozymes are the ones which are non-proteinaceous, and they are having a nucleic acid in the form of RNA. So except that one, all others will be having the proteinaceous nature. Here the proteins not only contains the protein part, but they also contains the non-protein part. So likewise based on the composition, so basically there are two types of enzymes are there. Those are simple enzymes and conjugated enzymes. So let us start with the first one, the simple enzymes. So simple enzymes are the one which contains only protein part. So that means here the non-protein part is not present in them. So this type of proteins are called as simple enzymes. Examples like in trypsin, pepsin, papain, amylase. All these are some of the type of enzymes where they are made up of only the protein part. And let us come to the second one, that is called as a conjugated enzymes. These are the enzymes which contains both the protein part and non-protein part. Here out of these two parts, the protein part is there which is called as apoenzyme, and the non-protein part is called as a cofactor. So here a conjugated enzymes means apoenzyme plus cofactor is equals to an conjugated enzyme. So this is otherwise called as holoenzyme also. So here holoenzyme is a functional unit of enzyme, and out of these two, so here these two are functioning in coordination with each other. That means in absence of cofactor or in absence of apoenzyme, the conjugated enzyme will not perform the catalytic property. That's the reason why they are always will be in the coordination cooperative function will be there. And let us see the individual parts which are present in them. The starting with first one, the cofactors. So here there are two types of cofactors are there. Those are, one is metal ion cofactor and second one is called as a organic cofactor. So here the non-protein part, based on the nature of the non-protein part, they are of metal ion cofactor and organic cofactor. So here if the cofactor is an metal ion, then we call it as a metal ion cofactor, and in absence of or without the presence of this metal ion cofactor or the particular ion, so the conjugated enzyme will not have that catalytic ability. So let us take some of the enzymes here, and otherwise we can call this metal ion cofactors as metalloenzymes. The other name for the metal ion cofactors are metalloenzymes. So let us take some of the examples here, that Mg+2 is acting as a metal ion cofactor for hexokinase enzyme. So as we know that hexokinase enzyme is enzyme which is participating in the metabolic functions. Then manganese, it is for IAA oxidase. And Fe+2, that is for catalase. So likewise Zn+2, carbonic anhydrase. Likewise many such metal ion cofactors are, that means without the presence of these metal ions, so that particular enzyme cannot participate in the catalytic ability, or it does not have the catalytic ability. So this type of nature will be there, and these cofactors are always tightly binded to the apoenzyme part. They are tightly attached to the apoenzyme part. Now let us come to the organic cofactors. Here there are two types of, and here these organic cofactors are the one which are made up of organic compounds, and these are of two types. Those are called as coenzymes and prosthetic group. Coenzymes are the organic molecules which are loosely binded to the apoenzyme part, whereas prosthetic group is the one which are tightly binded to the apoenzyme part. Of course, here we can say that the cofactors, in the cofactors, that is metal and cofactors, these are also tightly binded, but these are inorganic compounds. So here the prosthetic group is also tightly binded, but it is an organic compound. And here these are derived from the water soluble vitamins. The organic cofactors are derived from the water soluble vitamins. Like, let us take some of the examples, like coenzyme A, which have been derived from pantothenic acid, that is vitamin B3, and FAD, flavin adenine dinucleotide, that is FAD. This have been derived from flavin or riboflavin, that is vitamin B2. Some other examples are pyridoxal phosphate, which have been derived from pyridoxine. In the same way, thiamine, which have been derived from thiamine, that is B1 vitamin. So likewise, they have been derived from the various types of water soluble compounds. And let us see the next one, prosthetic group. So as we have already discussed, that prosthetic group is the one which is made up of organic compound, and that is tightly binded to the apoenzyme part. Like the heme group or heme moiety is a prosthetic group which is tightly binded to the apoenzyme part. So in the heme moiety, heme is a prosthetic group which is half part, or the which is tightly binded to the apoenzyme in the peroxidase enzyme. And apart from that one, there are some enzymes like FAD and FMN, that is FAD means flavin adenine dinucleotide, and flavin mononucleotide. So these two are having both the abilities, that is they can act both as a prosthetic group and also as coenzymes. This dual ability is there with the FAD and the FMN. So these are some chemical nature related to the enzymes.